Hi everyone, it's Mr. Sinti, and today I'd like to discuss one of the more intriguing and critically important concepts in the whole conversation about how cells communicate. Cells often communicate with one another by secreting chemical messengers. And so these chemicals are either floating in the blood and are perceived by cells, or if you're unicellular, they could be floating anywhere. They could be floating in a lake or someplace like that. And so I want to get into a conversation right now about how these chemical messengers are received. And so the reception is like the first of three steps in terms of what scientists call signaling transduction pathways. And so signaling transduction pathways is all about reception is part one, and then the transduction pathway is part two, and then a response is part three. And so this particular video is going to look at the details of reception. And so I kind of want to start off with looking at this baseball glove here. And so you might recognize the fact that a baseball glove, which is produced out of leather, can be a really good shape for catching things like baseballs. And so in the pocket, or this sort of alcove area right here, or the groove. And so what I'm going to be discussing is how there's molecules embedded in the plasma membrane of cells that are often protein, and that these proteins are going to be binding signaling molecules. And these molecules, these signaling molecules, are called ligands or ligands, depending on how you want to pronounce it. I'm going to call it a ligand. And so this ligand is like the baseball right here. And so what's fascinating is that, unlike, this is where this analogy kind of breaks down, unlike a baseball glove that could probably catch softballs or oranges or anything that's sort of circular, or even not, a receptor protein embedded in the cell membrane is extremely specific. And so therefore, messages being sent in the body through the bloodstream, for example, if it's a hormone, are able to go to particular target cells. And so specificity is really critical in reception. So there's a particular recepting protein for every ligand or ligand. And so the shape of that particular protein receptor is what's going to initiate the whole transduction pathway. And so let me just simplify it, like sort of the membrane like this, phospholipid bilayer. What I'm not drawing is this, now I am drawing it, phospholipid bilayer like this. And so these proteins are embedded like this, okay, so this receptor protein. And when a ligand comes and binds to it, if you're familiar with protein tertiary conformation, usually when something bonds to the protein, it will cause a shape change to take place. And so when the chemical binds to it, it therefore causes some kind of conformational, exaggerated, some kind of conformational change to occur. And this is the point. The chemical itself, especially if it's a water-soluble kind of larger molecule, it's not going to be able to pass through the phospholipid bilayer. And so when it binds to the receptor protein, what it can do is cause a conformational shape change, okay, in the receptor protein. And so these plasma membrane proteins will have a conformational shape change. So a shape change will result as a consequence of the ligand binding. Okay, so that's important. So here's an example. I drew this time the phospholipid bilayer, and here's one of the receptor proteins. And so if this is a ligand molecule that's attaching here, right here, because of the fact that it's chemically bonding, temporarily, it's probably some sort of weak bond. But what that's going to happen is it's going to cause this region down here to have a change. So a change in the shape down here. And if this particular part of the protein changes its shape, it thus allows it to do something or to do work. Some kind of physical movement is what we're talking about, a physical movement. And so this will allow this particular protein to change, if you will. And so that will allow the transduction of that signal message inside of the cell. This is obviously, or maybe not, outside the cell and this is inside the cell. And so this is what we're talking about. So maybe this ligand is floating in the bloodstream. So here's the blood out here. And so it comes, and this is the target cell, because this particular receptor protein has only eyes for this, as opposed to other signaling molecules that are traveling by that are different in shape. Okay, so this receptor protein is going to pick up this ligand, and then it's going to change shape. And so that's the most important point that I want to make so far. So these particular molecules, these signaling molecules, are water soluble, and so they can't really move across the plasma membrane. And so these receptors sort of span the plasma membrane, as I was drawing right here. Spanning meaning like crossing the phospholipid bilayer. So there's an outside and an inside part. And so I want to discuss in this video 3 of the major types of membrane receptors that are important in signaling and in signal transduction. And that is G protein coupled receptors, or sometimes called GPCR, and then receptor kinase, tyrosine kinases, and then simply ion receptor channels, ion channels. And so let's first start off with the G protein coupled receptor example. And so these particular G protein coupled, let's get into that for a second. The fact that it's called G protein means the fact that this particular receptor protein is coupled with, not shown, but I will draw it in, coupled with a protein that's on the inside. Again, this is on the outside of the membrane, and this is on the inside. This particular protein is called a G protein. And so that's why it's called G protein coupled receptor. So this is a G protein coupled receptor. Now, there's literally thousands of different kinds of G protein receptors. They do so many things, and they pick up different signals. And as it turns out, they're critically important. And just to throw out an example of why you would consider them important, about 30 to 50% of our drugs that pharmaceutical companies are producing are trying to target these receptors and try to fight off particular ailments. And some of the diseases that plague us, like, for example, whooping cough or botulism, these kinds of diseases that are caused by bacterial infections, sometimes those bacteria are producing toxins that interfere with the G protein coupled receptors. And so I wanted to emphasize that. And so what you're getting in this picture right here is that you can see kind of a detail of the phospholipid. That's what this is over here. And also what you're looking at is these sort of things that look like blue ribbon are the secondary structure of a protein. You might be familiar with the fact that this is an alpha helix. And so as it turns out that these proteins have 7 strings of alpha helix, and this particular molecule is cholesterol, sort of maintaining fluidity in order for this receptor protein to change shape. And so basically you have 1, 2, 3, 4, 5, 6, 7. You have these 7 protein alpha helixes that are sort of spanning the membrane. And so this is a little bit better of a drawing here. The signaling molecule will come along. I've been drawing that in yellow, so I'll continue to draw that. It'll bind to a specific site. It doesn't always have to be over on this particular side. There's a lot of these different GPCRs in the body. And so when it does bind, it will actually cause a conformational change in the protein. And because this portion of the protein on the inside of the cell changes shape, it'll affect the G protein that's associated with it. And so let's get into that. So if I were to attempt to draw that, let me see if I can do that. Let me go wide on this. So let me draw the phospholipid bilayer a little bit. 1, 2, I'll do a couple of them here. Here's the phospholipid bilayer, attempting to draw. Okay. And then let me draw this part of the 1, 2, 3, 4, 5, 6, 7. Okay, and then let's draw a little hook right there. And then I'll draw a little bit more phospholipid bilayer. Okay, let's do that. Let's do that like this. And so when the ligand comes along like this, keep it yellow, and binds, it's going to cause the protein, the receptor, to change shape. And what's that going to do is that it's associating with this G protein. And so let's get into the G protein a little bit. The G protein happens to be made up of a heterotrimeric subunits, and there happens to be 3 of them. And so let me draw that in blue. The G protein is going to be in blue. And so it has this one protein right here is going to be gamma, and another one's going to be beta, and the other one is going to be alpha. Okay? So there's 3 subunits. The reason that it's called a G protein is that the alpha segment of the 3, the alpha part, actually has attached to it a guanine diphosphate, a GTP, a GDP, guanine diphosphate. And when it's in this conformation, it's referred to as inactive. So it's inactive when a guanine diphosphate is attached to it. But what's interesting is when the ligand attaches, so this is step 1, when the ligand attaches to the receptor protein, it's going to cause a shape change, which is then going to change the shape of the G protein. And this whole thing is the G protein. This whole thing is the G protein. And so what's going to happen is it's going to cause the guanine diphosphate to be released, and it's going to allow a guanine triphosphate to be attached. And when guanine triphosphate attaches to the alpha subunit, it disassociates from the gamma and the beta, and it begins to move laterally across the phospholipids, because this is a fluid and it's attached. And so it's capable of, the alpha unit moves along, and you're like, well, where is it going? And so it moves along over here. This is the alpha right here, and it has the guanine triphosphate. Well, let's take a look at that. It's moving in this direction. And so the membrane protein receptor is associated with the G protein, as I was talking about. And when the guanine diphosphate is attached to it, it's inactive. But it can become active when a signaling molecule attaches to it. Okay? And so what's fascinating is when the guanine triphosphate attaches to the alpha subunit, the alpha subunit is capable of migrating along the plasma membrane, and it reaches a protein that's adjacent to it, and it's often an enzyme. Now, there's many different types of signaling transductions, but it's often an enzyme, and it's often inactive. And as you can see here, when the G protein, in particular the alpha subunit, has the guanine triphosphate, when it attaches to that enzyme, it changes the enzyme's conformation so that the enzyme becomes functional. So in other words, the active site is now available, and this is not showing detail. This is now available for many steps to occur, and transduction can occur inside the cell, and the cell will actually respond to whatever that message is. And when it's over, it's the opposite. The guanine triphosphate the enzyme loses a phosphate, it becomes GDP. The GDP then is attached to the alpha subunit, and then the G protein is inactive. So basically, the signaling molecule activates the G protein, which then activates an enzyme, and then things happen in the cell. So that's kind of generic, so I thought it might be productive to get into an example of that. And so if you're familiar with the fight or flight response, in other words, if an animal is in some kind of harm's way, like if you saw the video that I produced on cellular messaging, I was referring to an example of a cheetah chasing an impala. That will get your adrenaline going right there if you're an impala. And so basically that molecule, epinephrine or adrenaline, is a water-soluble hormone, and it's not going to be able to pass through the cell membrane. So what it does is that it binds to a G protein coupled receptor. What that does is change the shape of the protein, which then allows the G protein to lose its GTP, and then ATP is added, and then that migrates over, and the particular enzyme that it activates is adenylate cyclase, and that molecule adenylate cyclase is capable of doing something. And you're like, okay, you mentioned that, but what is it that it does? Well, what it does is the active site will convert adenosine triphosphate. And just to pause here for a second. Now, the detail of this is somewhat important, or else I wouldn't mention it. So it's like, okay, would I really require to remember this? Or the thing is, it's not about remembering it as much. It's like if you spend time with this, certainly you'll remember it, and you can look something up and pause the video if you'd like. But I think there's great beauty in the detail of this, and so go with me on this. So here's adenosine triphosphate. What that enzyme is capable of doing is converting it by removing two of its phosphates, something called a pyrophosphate, and producing adenosine monophosphate, cyclic, or what's known as cyclic AMP. Now, just to pause here for a second, just to give you a visual of what this is right here, ATP being converted to cyclic AMP. That enzyme right here, shown now in just green, it's a big protein though in the membrane. It can take ATP, so you might be familiar with ATP. It's adenosine triphosphate. Here's the ribose sugar. Here's the nitrogenous base adenine. Here's the 3-phosphate groups. So what it does is that enzyme rips the 2 phosphates off, so it's monophosphate, but then the ribose sugar connects this way, so it's cyclic. See that little cycle right there? Cyclic AMP. Now there's another enzyme that if you add water, it's called phosphodiesterase. You could turn it into just simply AMP, adenosine monophosphate. But see this cyclic AMP right there? This cyclic AMP is referred to as cyclic AMP, a secondary messenger. And the reason that it's a secondary messenger is that when this is all happening, when epinephrine comes to the cell, let's say it's in your muscle. Okay, so this is a muscle cell and it's receiving epinephrine. What it's going to do is it's going to cause cyclic AMP to rise. The concentration of that's going to rise, and then this is what's going to get stuff done. So this is the transduction inside the cytoplasm. What it does is that it activates a protein, and there's often more steps than this. This is still a simplification. A protein kinase, which then transfers a phosphate onto other molecules and gets things done. So if this is a muscle cell, what it might be doing is taking the energy that you find in the polysaccharide glycogen and produce glucose from that. Or if this is a blood vessel cell, it's causing the cell to dilate to allow more blood to come to the particular muscle, or raise blood sugar level, or increase heart rate. These are all the cellular responses to epinephrine. So that's what this is. And so cyclic AMP is getting that done. And so let me just pause this for a second and come over here. And let me show you an example of what I'm talking about here. In terms of, here's the epinephrine binding to the G protein cell receptor. Here's the G protein, and already the alpha segment is being activated. Let me let this play.
The G protein subunit, which dissociates from the others, carries a GDP, which is replaced by GTP when the subunit is activated.
Okay, now it's activated.
The activated G protein subunit then diffuses within the plasma membrane until it encounters adenylyl cyclase, a membrane enzyme that is inactive until it interacts with the G protein subunit.
And you're like, well, what's that going to do? Well, that's going to turn ATP into cyclic AMP.
When activated by the G protein subunit, adenylyl cyclase catalyzes the formation of CAMP from ATP. The CAMP formed at the inner surface of the membrane diffuses within the cytoplasm, where it binds to and activates protein kinase A, an enzyme that adds phosphate groups to specific cellular proteins.
Okay, so this enzyme, which is present all the time, this kinase, now it's activated. So it's an enzyme that's present, it's inactive, because you don't want it to be active, And when cyclic AMP binds to it, it now becomes active. It's active. It's active. It's doing things. And like, what is it doing? It's transferring negative phosphate groups. Let's see if I can write on top of this. This thing's going to be transferring phosphate groups, which are negative. Now that it's activated, it's a kinase that's activated. What do kinases do? They move phosphates onto other molecules. So let's take a look. Let's continue that.
In liver cells, protein kinase A phosphorylates and thereby activates another enzyme called phosphorylase, which converts glycogen into glucose 6-phosphate.
Okay, phosphorylase, again, sorry for the detail, but there's coolness here. Phosphorylase is then activated. And you're like, okay, now I am activated phosphorylase. What do I do? Well, it's basically like a pair of scissors. It's going to hack glycogen up into little tiny glucoses. And these glucoses, sugar, monosaccharide, they have phosphates onto them, either in the 6 carbon or the first carbon. So it's glucose 6-phosphate or glucose 1 phosphate. But eventually, this is going to produce glucose. And what's the cell going to do with that? Well, it could use it to burn it in glycolysis, and then the mitochondria will produce more ATP for energy because it's epinephrine and you want energy. Or glucose can simply diffuse out of the cell and then raise the blood sugar level because the animal's in jeopardy. Okay, so that's pretty interesting, right? That's pretty interesting. So I just wanted to show you that example. And so I mentioned at the start of the video, there was another type of receptor protein, and it's called tyrosine kinase. Now, these particular membrane receptors are kind of similar. They're proteins, but they have a lot of tyrosine, which is a type of an amino acid, attached to them. And I'll show you a picture of it right here. So it comes in two strands, and so these are monomers, and they'll come together to form a dimer. And so they're called tyrosine because they have a lot of this amino acid complex. So when the molecule comes and it binds to it, that's what we're talking about. And so one of the main differences is that the G protein only usually forms one signal transduction pathway, whereas these tyrosine kinases, when they attach, when the signal message attaches, these things form a dimer. They come together, and then ATP is then added to the tyrosines, and it becomes active. And you're like, well, what's that going to do? That then transfers phosphates to other proteins in the cytoplasm, which forms other cascades that get a lot of things done. And so this really amplifies the cell's ability to produce something. So in other words, an example of this would be growth factor. So if a growth factor comes, then it'll tell the cell to initiate the cell cycle, especially into the mitotic part of this. Now, this is good when it works, and this is troublesome when it's not. If there's a lot of these receptor tyrosine kinases in the membrane, abnormal amount. Why would there be an abnormal amount? Maybe the cell has aneuploidy in there, and there's more of those chromosomes, which are making more of the receptor proteins. And if there's more receptor proteins, you're going to be picking up a lot of signals to divide more often than normal. So abnormal functioning of these kinase receptors are associated with a lot of cancer. Not only do they pick up the signals to divide, but they start producing excessive amount of protein, which then throws off the cell's ability to regulate its division, such as the production of cyclin, which is a critical regulating protein in mitosis. Now, the third example of a receptor protein is simply a ligand-gated ion channel. Now, ions are small, but they're electrically charged, and they're not getting through the cell membrane. And so what's interesting is I'm going to give you an example of either a sodium or a potassium, or sodium or a calcium ligand ion channel. And so let me set the stage. These ions cannot get through the cell membrane. And so what can happen is the ligand or signaling molecule, like maybe it's a neurotransmitter, that attaches to the ion-gated receptor, which causes a change in the shape of the protein, which then allows the ions to open. So it's a chemically gated, like a gate that swings open and close. It's a chemically gated ion channel. So when the ligand's present, the chemical, the channel is then open and then the cell does something. And when the chemical is not present, the gate closes. So that's kind of an easier, but this is critical. This is often how neurons function. And so finally, let me get into a discussion with you with regards to if a chemical is lipid soluble. If a chemical like a hormone, like a steroid hormone, is lipid soluble, that can actually just go right through the cell membrane, the phospholipid bilayer. But it still needs to encounter a receptor protein. So these are intracellular receptors, not in the membrane. So these intercellular receptors are found either in the cytoplasm, cytosol, or in the nucleus of the target cell. And so these things come across and then they bind with the receptor. It activates the receptor, and then that usually goes into the nucleus. It binds to DNA, and it gets the DNA in a particular gene to become active, which then produces transcription, which then makes messenger RNA, which then produces proteins, which gets things done. So here's an example of a hormone, testosterone, male hormone, that then just simply goes across the cell membrane. It does interact with an intracellular receptor protein, and then that activates it, and then that goes maybe into the nucleus, and that interacts with a particular gene, and then these genes are then transcribed, and then proteins are produced, and that's the response. So that's pretty cool. So messenger RNA is produced, and then a new protein. So this is the response. And so an example of that, just so that it's crystal clear, would be like a steroid hormone I was mentioning. Let me let this play.
Steroid hormones are not water soluble. They travel in the blood attached to protein carriers.
So this is
when steroid hormones arrive at their target cells, they dissociate from their protein carriers and pass through the plasma membrane of the cell.
Notice how there's no receptor protein. It just comes right in. Here's the receptor protein. It's intracellular.
Some steroid hormones bind to specific receptor proteins in the cytoplasm and then move as a hormone receptor complex into the nucleus.
Like testosterone.
Other steroids travel directly into the nucleus before encountering their receptor proteins, not shown.
No problem.
The hormone receptor protein, activated by binding to the hormone, is now able to bind to specific regions of the DNA. These DNA regions are known as the hormone response elements. The binding of the hormone receptor complex has a direct effect on the level of transcription at that site. Messenger RNA, mRNA, is produced, which then codes for the synthesis of specific proteins.
All right, so there's an example of an intracellular receptor protein. So we've gone over the G protein receptors, the tyrosine kinase receptors, the ion ligated receptor, and now intracellular protein receptors. I hope this video was helpful in understanding the very important concept of how receptor proteins are important in signaling, capturing signal messages from cell to cell, and are the first step of signaling transduction pathways. Thanks for watching.